Hi, so in this podcast, we're going to look at the monohybrid crosses, and this will also be a podcast on how we would go about doing Punnett squares or answering genetic problems for monohybrid crosses. So this takes us through some classic monohybrid crosses that Gregor Mendel did. And the first monohybrid cross that Gregor Mendel did was the P1 monohybrid cross. Remember from the last podcast that P1 stands for parental. And so one of the traits that Gregor Mendel used was seed shape. And the alleles for seed shape are round, which is dominant, and wrinkled, which is recessive. And what Gregor Mendel did was he crossed round seeds and wrinkle seeds, and he made sure they were purebred, they were true breeding. And so in any Punnett square, in any genetic problem, the first way to start going about this is to establish your parentals. So once you've identified your parentals, and I would simply write them out directly over top, you can then start your Punnett square. Now a Punnett square is a tool, remember, that we're going to use to answer genetic problems. Remember that each of these letters stands for one of the chromosomes and one of the alleles on the chromosome. And during meiosis, those alleles separate from each other. And so this is what a Punnett square sort of represents. The chances of, or during meiosis, when alleles separate, the possible offspring combinations that we could get from fertilization. And so if we were to do this Punnett square, we would put one of the parents on the top, one of the parents on the side, and then we would simply fill in the boxes. Okay, and this tells us the possible offspring. So when Gregor Mendel did this and he crossed round and wrinkle seeds, he found that his genotype, or he found his phenotype to be 100% round. The genotype of that, and he would not have had the ability to look, although through math he actually did or could figure this out, was 100% heterozygous. Now, the other two things that we sort of look at are the ratios. So the ratio for the genotypes is simply just one. There's only one possible genotype. And the phenotypic ratio in this case is also one. There's only one possible genotype. So just as a review, when he crossed a homozygous dominant and homozygous recessive, all the offspring were hybrids, they were heterozygous. The offspring in this case are called the F1 generation, remember? And the genotypic and phenotypic ratios is they're all alike. The next cross that Gregor Mendel did, because he asked the question, well, what happened to that? What happened to that other trait? What happened to that wrinkled trait? So he took offspring of the F1s and he allowed them to self-pollinate. In other words, he crossed the F1s. So again, these are the parents, the F1s that we're looking at. Constructing a Punnett square for this through a monohybrid cross. And in any genetic problem, you will have to construct Punnett squares as part of the question, in order to get full marks and also in order to help figure out the possible combinations. And that's sort of what he found there. So if we look again at the genotype, in this case, there were three possible genotypes. 25% of the offspring were homozygous dominant, 50% of the offspring were heterozygous, and 25% of the offspring were homozygous recessive. Phenotypically, though, what Gregor Mendel would have seen initially after he did the experiment was that 75% of the offspring were round seeded and 25% were wrinkled. Okay, notice that when two heterozygotes are crossed together, we end up with a little bit different numbers. Genotypic ratio in this case is 1 to 2 to 1. There's one homozygous dominant to two heterozygous to one homozygous recessive. Okay, to show you that on the Punnett square, there's one here to 2 to 1 there. So a 1 to 2 to 1 ratio. Phenotypic ratio, there are 3 dominant to every 1 recessive. Okay, again, quick review. When we cross heterozygous with heterozygous, we get 25% homozygous dominant, 50% heterozygous, 25% homozygous recessive. These offspring are called the F2 generation. The genotypic ratio is 1 to 2 to 1. The phenotypic ratio is 3 to 1. So just as a quick diagram to show what these seeds look like, here's what it would look like in the actual cross that Mendel did. Some of the seeds would be round like this one, and some of them would be wrinkled. And this is what he did. He counted thousands and thousands of offspring to come about to his 3 to 1 ratio. And every time he did it, he got that sort of a ratio. The next thing, or the last thing that Mendel did, was he did a test cross. So with his dominant traits, he didn't know which ones were homozygous dominant and which ones were heterozygous. But he developed a method called a test cross, in which he would cross a pure and a hybrid from his F2 generations. So this is known as the F2, or the test cross, the F2 cross. There's only one possible test cross, so we can only cross the homozygous recessive with some sort of a known hybrid or with a known dominant, and that would give us our results. So for a test cross, we only use the homozygous recessive. And this is what it looks like. So again, if we're using wrinkled seeds, if we have a known dominant, let's say that one was, for example, a heterozygous, this is what it would look like. So again, running our Punnett square, setting it up. We would see that there are, genotypically, there's 50% big R, little r, 50% little r, little r. The phenotype with this would be 50% round, 50% wrinkled. The ratios would be a 1 to 1 and a 1 to 1. Okay, and that's if it was a heterozygous. But let's imagine for a second that the plant that we were trying to figure out was homozygous dominant. And let's just run a test cross to show the two differences and why we use the homozygous recessive. So let's say that that was this, and we were to run our Punnett square with a known dominant that just happened to be homozygous dominant. What we would see is, going back to our very first parental cross, we would have 100% round offspring, a genotypic and phenotypic ratio of 1. So this test cross, crossing this homozygous recessive, allows me to see whether or not the plant that I'm looking at is heterozygous or homozygous dominant, because remember, phenotypically they'll look the same. So that's sort of the basics to a test cross. If we review it, this is sort of the basics to it, the review part, crossing the recessive with the heterozygous or the hybrid. And what we get is, when we do that and when it is a heterozygous, we get 50% homozygous recessive and 50% heterozygous. So in other words, the phenotypic ratio is 1 to 1. We call this a test cross because the offspring have the same genotypes as the parents do. What I want you to do now is to go through the different crosses if you need some practice. Work through the P1, the F1, and the F2 crosses for each of the other 7 plant traits. And then remember, there's also the genetics problems that I'd like you to complete on the monohybrid cross.